Hello and welcome to Reproduction 1. This is a review of the cell cycle and also a review of a term called ploidy. So if you look at the right hand side of the cartoon, hopefully you'll recognize that that is DNA with its base pairs, A pairing with T, C pairing with G, and it's showing the DNA in the process of DNA replication. So the mother strand is being duplicated into two daughter strands to make two new cells. On the left side of the cartoon, that top circle, it says 46 2n/1x. That means in the human there's 46 there's 46 chromosomes, and when there's 46 chromosomes present in a cell, that's referred to as 2n, or 2 times the ploidy of that cell. And then I've got not just a 2n, but I've also got a 1x. So the x denotes for me if the DNA is duplicated or not. So in the top circle there's 46 chromosomes, 23 from the mother, 23 from the father. That's the 2 part, the 2n part. And then the 1x means that those chromosomes have not yet been duplicated. And then if you look at the sphere just below that, it's got 92, the number 92. That means the chromosomes, instead of 46 chromosomes, the 46 chromosomes have been duplicated. So now you have 92 chromosomes, and that's where you get the 2x in the 2n, 2x. And then after the process of mitosis, then you have, from one mother cell, you have 2 daughter cells, and both the daughter cells are returned to having 46 chromosomes each, 23 from mom, 23 from pop, and the chromosomes are not duplicated, thus it's 1x. So I told you ploidy then can have two meanings. Ploidy in terms of genetic content. In a diploid cell, in a diploid human cell, our ploidy is 2n. That means you have 2 of each gene if you're diploid, and you only have 1 of each gene if you're haploid. If you look at ploidy in terms of chromosome number, then you can refer to that as a C or an X. I call it an X. Some people call it a C. That's just the total amount of DNA. Here's a cartoon of the cell cycle. You can see that that right in the middle there's a green arrow that kind of goes around the circle. It's called interphase. And then there's a yellow piece of that disc that's called the mitotic phase. And then it's got a couple cartoons on the outside. At the top, it's got a cell that has not yet been duplicated. And then in the bottom, the 2 circles outside the cell cycle cartoon, at the bottom you've got the chromosomes lining up. And then at the very tip of the yellow arrow, you see the chromosomes have split, and this is in the process of making 2 new daughter cells. So if we go back to the colored circle, like I said, on the outer rind of that circle you have interphase. Most cells spend their life in interphase. Within interphase you have G1 of interphase, S of interphase, and G2 of interphase. Within mitosis you have prophase of mitosis, metaphase of mitosis, anaphase of mitosis, telophase of mitosis, and then you have cytokinesis. And we'll go over each one of these. When you're in G1, the G1 phase of interphase, you're just doing your regular job. You're just living as a normal cell. In the S phase, that's when you replicate your DNA. So that's where you go from having 46 chromosomes to 92 chromosomes that we talked about in the previous slide. If you are in G2 of interphase, then you are still doing your job to maintain yourself as a cell, but you're preparing yourself for division, and that is the mitotic phase. In mitosis, the chromosomes will divide equally, and then for the two daughter cells to actually separate, that's called cytokinesis, when you divide the cytoplasm and the two cells then become separate. Interphase, G1S and G2 of interphase represent about 90% of a cell's life cycle. Some cells are stuck in this subphase. It doesn't show it on this cartoon, called G sub O. Like neurons, they don't divide. So let's go over these in a little more detail. Interphase of the cell cycle. Let's look at G1S or G1. So on the right side of the cartoon, we've gone over that, or excuse me, on the right side of the slide, that cartoon we've gone over. Now this is a new cartoon on the left side. It shows a cell, and in the middle you've got the nucleus with the chromatin in it. It says chromatin is duplicated because the chromosomes have duplicated, but you can't see the chromosomes clearly yet. They call it chromatin. And then you've got these things called centrosomes, and that's a space in the cell with these proteins called centrioles, and the centrioles are going to ultimately be there to pull the cell apart into two new daughter cells. So now again, on the right side of the slide, we're in the mitotic phase, so we're in that yellow part of the cell cycle diagram. So we're going to look at prophase and then also pro-metaphase. It isn't written as pro-metaphase, but some books say there's a prophase of mitosis and a pro-metaphase of mitosis. So if you are in the yellow part of that diagram on the right-hand side, you're in mitosis. That means you've already duplicated your DNA. When you duplicate your DNA, but the duplicated DNA is still stuck together, they call that a sister chromatid. So if you look at the left part of the screen, so the left cartoon, you can see that you've got two cells. The cell on the left has got the duplicated DNA in sister chromatid form, and now where the sister chromatids are joined, that's called a centromere. Now don't get the centromere of the sister chromatids confused with the centrioles that we see just above the nucleus in that diagram. So the centrioles, then those yellow things, that the centrioles on the left side of the screen in the left cell are those yellow entities, and then they've got spindle fibers, those yellow lines coming out of the centrioles. Those are called spindle fibers, and they're ultimately going to help pull the cell apart. In prophase and prometaphase, then you can see that the sister chromatids are lining up, and the nuclear membrane, that purple thing, has disappeared. You can also see that the early mitotic spindle has progressed. If you look at the cell in the middle of the slide, the mitotic spindle has progressed, and pieces of the mitotic spindle have actually attached to the centromeres in the sister chromatids, and they're calling it in this cartoon a kinetochore. So where the mitotic spindle is attached to a kinetochore, the kinetochore is the protein structure within the centromere of the sister chromatid. So now let's look at metaphase of mitosis. So again on the right side of the slide, you can see we are in the mitotic phase. That's the yellow part of the cartoon. We're in metaphase of mitosis. And now look at the left-hand side. You can see the cell with the sister chromatids lined up on what's called the metaphase plate. So we're in metaphase. And the centriole has sent out spindle fibers to connect to the kinetochores that live in the centromeres of the sister chromatids. And so the process is lining up these sister chromatids to be pulled apart into two new daughter cells. So each daughter cell will get an equal number of chromosomes. So as we progress into anaphase, we see that the sister chromatids are being pulled apart on the left-hand side of the cartoon. The purple sister chromatids are being pulled apart as the centrioles and the spindle fibers work their molecular magic. And then at the end of the mitosis, which is telophase, and then the beginning of cytokinesis, which is when the cytoplasm divides into two parts, equal amounts into the two daughter cells. So if you look at the far left part of the screen, you see an actual scanning electron micrograph of a cell beginning to form, actually looks like 4 new daughter cells, but in mitosis you just form 2 new daughter cells. So if you look at the cell in the middle, you can see that you're beginning to form the 2 new daughter cells. Each daughter cell will have a centriole, that yellow thing. The spindle fibers are starting to disintegrate. And then there's, if it's a human cell, each daughter will have 46 chromosomes. And then cytokinesis ensues at the end of mitosis. That's when the cytoplasm splits, usually into equal size sets. But when we go over oogenesis, meiosis, we will see that when you make an egg, only one egg will get all the cytoplasm, and the other daughter egg will not get any of the cytoplasm. But we'll do that when we talk about meiosis in females.